Morning, my darling, I'm telling you this to let you know that I'm sorry you're sick. Though tears of sorrow won't do you no good, I'd be your doctor if only I could. What do you want from the liquor store? Something sour or something sweet? I'll buy you all that your belly can hold. You can be sure you won't suffer no more. I swim the ocean on the deepest canal.
Welcome to COVID Radio. We're here today with Ted Hawkins' hit Sorry You're Sick. And now we're going to take a break from our regularly scheduled songs and talk about the innate immune system. All right, so our stuff. What are we doing? Module. This time is about the immune system and integrated with that conversations about COVID-19. So I just finished recording with Misha and Laureen and several of you a conversation about COVID-19 and the immune system, and that by the time this gets up, that's certainly going to be posted. So make sure you watch that. There's some questions from your study guide about that, and it was a pretty interesting conversation, and both Misha and Laureen had some great things to share. So here's where we're headed. We're going to talk about, I thought we'd take a step back and just talk about the differences between bacterial and viral infections and how they infect the body differently. And then the big picture is you have this innate immune system and this adaptive immune system. Innate being kind of your general response to anything, any tissue damage or any kind of infection that occurs. And then your adaptive immune system is that specific ability to target a specific pathogen and kill it and get rid of it. Your innate immune system comes online pretty quickly, within minutes of an infection or a wound, whereas your adaptive immune system is going to take a few days to ramp up and get working. So within the innate immune system, we're going to talk about the inflammatory system. Under the adaptive immune system, we're going to talk about how the immune system gains its memory. We'll also be talking about how vaccines work. And then specifically, there are lots of different types of immune system cells. It's striking when you get into the immune system, just how complicated it is. It gets complicated really quickly. So we're going to focus on a few of those immune system cells, and one of them are called cytotoxic T cells, and they are one of the cells that are primarily responsible for helping you get rid of a viral infection. So we're going to talk about how those work. A little bit about allergies and autoimmune system. A lot of you had questions about those, how they work, what they do, what they're doing in the body. And we'll also be talking about ways to boost your immune system. Laureen did a tremendous job about that today, giving you some specific ideas about, in addition to some sleep and to managing your stress, things you could eat and supplement your diet with. And integrated with all of that is a conversation about COVID-19. So I've gone through all your questions. I farmed some out to Laureen and Misha, so some of them were talked about there, and then some of them I'll be talking about as we have our lecture together. So bacteria versus a virus. Things to know. One, bacteria are a living organism. They're a single-celled living organism. They're prokaryotes, if you know the difference between prokaryotes and eukaryotes. If you don't worry about that. But versus a virus, which is not a living cell. Viruses are a piece of genetic material. Sometimes it's DNA, sometimes it's RNA. DNA is the kind of genetic material you have inside of each one of your cells. And the way you use that genetic material is you make a copy of a gene and then use that copy to make proteins. We call that copy RNA. So some viruses are DNA viruses, some viruses are RNA viruses. And in the case of COVID-19, it is a virus that has a piece of RNA in the middle of it. And that's that little pink spring in the middle of that virus. And then around that virus is some kind of protective coat. And in the case of COVID-19, it's got a lipid membrane along with a bunch of proteins. And the great thing about having a lipid membrane as part of your coat is that means anytime you get soap interacting with that lipid membrane, it dissolves it. So because soap acts like a surfactant and it breaks up all those membrane tails that are keeping the virus together. So that's one of the reasons why washing your hands with soap, washing things with soap is so effective at killing this virus. As far as scale of things, bacteria are much larger than a virus. In fact, viruses can also infect bacteria. So some other things. Bacteria. Because they're a living cell, they can divide on its own. Doesn't not have to invade your cells. And it's also important to remember that most bacteria don't cause an infection, and they're actually very helpful to our health. Our gut flora plays a big role in helping us to digest foods. It helps to inform our metabolism. In fact, if we have a healthy diet and we have a healthy microbiome, it can help increase our metabolism. And those healthy microbes also help to teach our immune system what's what's a friendly bacteria that's helping us to survive, and what's a foe, what's a pathogen. But a few bacteria can cause diseases. And whether they're friendly bacteria or they're bacteria that are causing diseases, they're using your body for resources. So your body is providing shelter, so a place to live, as well as food. And the issue with bacterial infections is when they cause diseases, one of the things they can do is they can produce toxins that can be damaging to our cells and damaging to our bodies. And bacterial infections in general tend to last longer than viral infections, and we can use antibiotics to kill bacterial infections. Antibiotics do not work for vaccines. Vaccines, smaller protein or membrane coated genetic material, and they have to invade a host cell in order to reproduce. Humans can get viruses, any living thing can get a virus. Plants get viruses. In fact, it's tulip season, it's springtime. So if you're a fan of tulips, the ones with stripes in the petals, those stripes are actually caused by a virus. So viruses have been used in horticulture for years to change the look of different plants, the way the pigment's expressed in different plants. So the issue with a viral infection is they're not producing toxins, but they do cause cell death because they are getting into the cells, taking over the cell's machinery to make more viruses. Eventually, those cause cell death and cell damage. So how do we fight those infections, whether it's a bacteria or a virus? Well, we start out with your innate immune system, which is what this lecture is going to be about. The next lecture I'll separate them, is going to be about your adaptive immune system. But just to compare, a quick overview, compare and contrast. Your innate's more general. It's attacking anything that's in the body, and it's a lot faster. So your body can get that innate immune system going pretty rapidly. Versus your adaptive, it's target specific. So this is the one that's using specific antibodies to match antigens on a pathogen to target them and to get rid of them. But it takes days for that system to ramp up and respond to an infection. So here's an image from your book that kind of differentiates between your innate defenses and your adaptive defenses. And we'll be going through these different components. But your innate defenses are broken up into two categories. One, your surface barriers, which includes your skin and your mucous membranes. And then you have internal defenses. So if something gets past that skin or mucous membrane barrier, then you have a series of internal defenses. We'll be talking more about those that just generally attack and get rid of viral and bacterial infections. and also cause inflammation. And inflammation can help get the body ready for something like wound repair as well. And then the adaptive defenses. So this is your specific antigen antibody targeted immune system. And that is subdivided into humoral immunity, meaning your antibodies are moving around in the blood and used to target different cells, versus your cellular immunity, where we're targeting specific cells and killing them. So your surface barriers. So your first big one is your skin. Skin's great barrier from things as long as it's intact. If it gets broken, then of course not such a great barrier, but when it's intact, it's a great barrier from bacteria and viruses. Part two of your surface barriers are your mucous membranes. So we have this, right? We have these mouths and nose, and that leads to these tunnels inside of our body. And if you remember, right, our tube within a tube, that you can think of your body as this tube that's surrounded by this body. So that tube, you can think of that as your respiratory system, your digestive system, the urinary system, the reproductive system. So any system that has this outer opening to the external environment is a place where viruses and bacteria can get into the body and invade and cause infection. And so all of those are lined with a mucus, which is handy. Your mucus is made by, this image is showing you some goblet cells here that are within the epithelium of your mucous membrane. You also just have seromucous glands that are producing mucus. And what's great about mucus is it's sticky, so it helps to bind anything like a virus or bacteria that's invading your body. Mucus also is full of different proteins and other chemicals that can target and kill bacteria and viruses. We also have this acidic mantle on our skin, and and our urine is acidic. The vagina is acidic, and that acidic mantle helps reduce bacterial growth. That bacteria don't like to grow so much in acidic environments. And as well, furthermore, in our respiratory system, in your nasal cavity and your lungs, those are lined with cilia. So you may or may not remember from anatomy that your nasal cavity, your trachea, and your bronchioles are lined with ciliated pseudostratified columnar epithelium. Those cilia are handy because they're actively sweeping that mucus back towards your pharynx. So either from your nasal cavity into your pharynx, or up from your bronchioles and your trachea into your pharynx. And it's sweeping all of that mucus, all of that debris, all of those dead bacteria and viruses that have been killed into your mouth, where you can swallow them, and then usually your digestive system is able to digest anything else that's causing an issue. So table 21.1 lists some more detail, if you're interested in looking at it, some of the things that your skin and mucous membranes provide to you as far as defense. So we've talked about the acid mantle of the skin. You've also got this acidic environment in the stomach, and the acid mantle of the vagina. Cilia, these great nasal hairs, right? They're your first line of defense. They act as these little brooms catching debris for your body. So what if a microbe / irritant gets past this really great surface? The preview video I had you watch talked about a mosquito bite. Right? And you have witnessed that within minutes after a mosquito bite, you get this inflammatory process. It gets hot, it gets red. and your skin is responding to that invasion, whether it's a microbe or an irritant that gets left by something like a mosquito. So what does your body do then? Well, now we're into these internal defenses that are still innate. They're still general, they're not targeted, but they are effective. First of all, you have some cells. You have these innate internal defenses. Cells, yes, Erin, thank you for reading the slide for us. Phagocytes are a specific type of cell that are able to undergo phagocytosis. We've talked about these macrophages before when we were talking about atherosclerosis and the development of plaques. Neutrophils are also phagocytes. And so by phagocytes, what we mean is this is an image of a macrophage. So here would be the cell body, but it has all of these long extensions where they can actually reach out and grab something like a bacteria, pull it back into the cell body and digest it using lysosomes. We also have something called natural killer cells, which is a great name for things. And what they do, they don't do phagocytosis. They're not bringing in the bacteria or the virus into their bodies and digesting them, but basically they talk other cells into suicide, which we call apoptosis. So a natural killer cell, when it finds something like a bacteria, it connects with it through receptor proteins, and then they have this chemical conversation where basically the natural killer cells convinces these cells to go through apoptosis or cell death. And none of these three cells, the neutrophils, the macrophages, and the natural killer cells, that's none of these are specific. They're not targeting a specific antigen, but they're just able to detect some general abnormalities of the cells. So in this way, these cells can attack things like cancer cells that are abnormal in the body, as well as getting rid of bacteria. So here's an image from your book about how phagocytosis works. You would have the purple blob here is the phagocyte. So this works for whether it's a neutrophil or a macrophage. And they're going to bind on receptors, protein to protein receptors. They're going to recognize that this is just some kind of general pathogen that the body needs to get rid of. It's going to envelop it in this vessel. So this is surrounded by a plasma membrane. And here's our, let's call them bacteria. That's what they look like to me. And then that's going to fuse with this lysosome. And this lysosome has a few tricks. One is acid. So it's got this acidic environment, so it can start to dissolve that bacteria with acid. It also has digestive enzymes where those enzymes are able to destroy and digest the bacteria into smaller pieces. And then step 5 is optional. They may or may not go through exocytosis. So sometimes they leave them inside of the cell and sometimes they don't. Differences between the neutrophils and the macrophages is neutrophils are only good for a short period of time. They can only do limited amounts of phagocytosis, versus macrophages are voracious eaters. They can survive several days and eat many, many bacteria before they die. So one thing I wanted to talk about before we leave these innate internal defenses, and Misha talked about it briefly in our session earlier this afternoon, but one of the symptoms of COVID is feeling really bad. And by the way, when you read that most people will have mild cases of COVID-19, mild in the terms of a medical setting means that you don't have to go to the hospital and you're not on a ventilator, but it's still a really crappy virus. You're still, if you get that virus, you're gonna have a fever, you're gonna feel fatigue, you're gonna feel really crappy for a few days. So mild on the scale of things just means you're not in the hospital. But from the friends we've talked to, it's still one of the, basically the worst viral infections they've ever had in their lives. But it seems to, you feel really awful for a little while, and then you start to feel better, and you'll feel better for a day or 2 or 3, and then it comes back. And some people feel even worse than they did during the first round, and it's more intense. And one of the things they're finding that COVID-19 is able to do is somehow dampen down this innate internal defenses, and specifically somehow make it so these natural killer cells aren't doing their job. And so your body feels really bad, and then it kind of turns off this inflammatory process that we're about to talk about. And the virus is somehow evading and actually down-regulating these natural killer cells for a while. And while they're doing that, the virus is still reproducing and moving further and deeper into the body, which then can cause the cytokine storm that Laureen talked about, where your immune system is just in this positive feedback loop where more cytokines, which are pro-inflammatory chemicals, are being released, and that's causing the development of and the division of more immune system cells. And when you have more immune system cells, those are going to release more cytokines. You just get in this positive feedback loop of an explosion and an amplification of the immune response. I was on a roll. I forgot what was next that I wanted to say. Will the train come back? Maybe not. It'll circle back eventually, because you know it's a loop. So your internal innate system, you have these cells, right? Macrophages, neutrophils, natural killer cells that can either consume through phagocytosis or convince something to kill itself through apoptosis. And then that is combined with inflammation. So inflammation, here's another picture of inflammation. So guess which hand is inflamed? Red, it feels hot to the touch, you have swelling or edema, and it feels pain. And this response is localized. It's not throughout the entire body. The point of inflammation is well, let's see, look at the benefits. I have the next slide. So it's attempting to isolate the infection, and it does that as the swelling occurs. There's also proteins, exudate, being released that kind of forms this gel around the area that's inflamed. And that gel-like substance is going to reduce the ability of whatever is in that inflamed area to move to other places in the body. The inflammation also helps your body dispose of debris and pathogens by calling in those neutrophils and macrophages and natural killer cells. So that's part of the inflammatory process, is activating those cells and calling them in. It's also getting that body ready for repair. It's doing that because that inflammation means we're sending in more blood to the area. And with more blood, there's more oxygen, there's more nutrients, there's more materials for the body to heal itself from a wound. And it also alerts your adaptive immunity. So that specific immunity, it starts putting out the call like, hey, we're taking care here. We're in triage here with our innate internal inflammatory system, but we could probably use some backup. Things are gonna get worse. So we could use some help. So it alerts your adaptive immunity. So let's talk about what gets inflammation started. So damaged cells are going to call for help, and they're going to do that in a variety of ways. And And damaged cells could be damaged from a wound. They could also be damaged from some sort of pathogen infection because the bacteria are releasing toxins or the viruses are actually invading the cells. And one of the responses that occurs is there are these mast cells that release histamines. So this purple blob is representing a mast cell. And they do several things, as you can see, but the thing we're going to talk about right now is once they get alerted to damaged cells, they're going to start releasing histamines. And histamines are a molecule that really help to get the inflammatory process started. And what do histamines do? Well, one of the things they do is they dilate your blood vessels. That's handy because that's going to move more blood into the area. Now, histamines are not systemic. They are being locally released. So we've got local dilation of the blood vessels. That's part of what makes things red and hot because we're moving more blood into the area. Histamines are also going to cause the capillary beds to become more leaky, meaning that the tight junctions that are between the epithelial cells that line the capillary beds are going to loosen up a little bit. They're going to get leaky, and now we're going to start getting fluids from the blood plasma into the area. And so it's going to start to swell, or we're going to see edema. And it's thought that swelling could be putting pressure on nerves, and that could be part of what causes pain. Part of the pain could also come from toxins from bacteria, and it could also have other sources. There are also chemicals that get released that make your pain receptors more sensitive during the inflammatory process. And then what else do histamines do? They're very, very busy. They're also going to start to call in the phagocytes. So things like the macrophages, and these here are the neutrophil cells that are going to come in and perform phagocytosis of the pathogens. So histamines, super important in getting the inflammatory process started. Another component of our innate immune system are some chemicals in the body, one of which I want to talk about, and that is called interferons. And their job is to reduce viral infections. But at this point, I thought it might be very valuable for us to just talk about in general what causes how do viruses invade our body in the first place. So let's do that. Keep your eye on the dot. Does it seem like zombie music to you? Thanks.
Contact. This is the moment when infection begins. How does infection happen? You see the tiny shapes there on the virus particle surface? They seem like random blobs. In reality, they are tiny keys, proteins that have evolved to the perfect shape and size, enabling them to unlock these receptors on the surface of this lung cell. Like a smile and a handshake, the virus deceives this lung cell and is welcomed in, where it will command the cell to make more virus particles.
Now we're going to follow that up with, once the virus gets in the cell, what does it do? Perhaps I have a virus on my computer. Let me pause you and see what I can.
Every cell in your body is a tiny factory, constantly making proteins. Your genetic code, or DNA, directs every activity. Protein-making machinery in the cell reads the cell's own DNA and makes sure the cell proteins go where they're needed. Viruses have their own genetic code, but they don't have the machinery to copy their DNA. So viruses need your cell's machinery to reproduce themselves. A virus starts this process by using the keys on its surface to gain entry. Like a Trojan horse, it enters the cell. The virus releases its own DNA into the unsuspecting cell and hijacks the protein-making machinery. Now, this vital cell function becomes an assembly line for new virus particles. No longer in service to your body, the cell now serves the virus, manufacturing thousands of new virus particles, which are released to enslave more cells, thousands and thousands more.
It really does seem like the zombie apocalypse. Huh? And of course, those cells would not be able to do their normal functions, but would just be making more viruses. And eventually those cells die, either because they're exploding because they have so many viruses in them, or your immune system actually kills them. So you have this marvelous protein called interferons, and they help reduce viral infections. But before we get there, looks like I wanted to talk about COVID-19. I remember now. The specific lock and key receptor for COVID-19 is the ACE2 receptor. And the ACE2 receptor is part of, remember the renin-angiotensin sequence that we've talked about that helps regulate blood pressure? Well, the ACE2 receptor is part of that whole cascade, and that is the specific protein that these viruses are using to dock. And that seems to be especially effective inside of the lungs. And those ACE2 receptors are on a specific type of cell in your lungs. They are on the type 2 alveolar cells of the lungs. So remember those lungs that are making their surfactant to help prevent your alveoli from collapsing when you're breathing in and out? Well, those are the cells that COVID-19 is invading. And
the protein that the coronavirus uses to attach to human cells has a compact ridge that allows it to attach more strongly to human cells than similar viruses, allowing it to infect better and spread faster. The new coronavirus, SARS-CoV-2, attaches to human cells through what's called a spike protein. After the spike protein binds to the human receptor cell, a protein on the cell surface that serves as a door into the cell, the viral membrane fuses with the human cell membrane, allowing the genome of the virus to enter the human cells. All coronaviruses attach to human cells through spike proteins, but different coronaviruses have spike proteins with different structures. In February, a group of researchers at the University of Texas at Austin and the National Institutes of Health mapped out the molecular structure of the new coronavirus's spike protein. Now, another group of researchers used X-rays to further explore the coronavirus's spike protein and the human cell receptor it binds to. Their goal was to understand why the coronavirus's spike protein is so good at infecting cells compared with similar coronaviruses known as SARS-CoV that caused the severe acute respiratory syndrome, SARS, outbreak in 2003. Both SARS-CoV and SARS-CoV-2 bind to the same human receptor known as ACE2. They found that a few genetic mutations led the SARS-CoV-2 spike protein to develop a more compact molecular ridge than that of SARS-CoV. This more compact structure and several other small differences allow SARS-CoV-2 to attach more strongly to the human ACE2 receptor, thereby enabling it to better infect cells and thus spread faster than the SARS coronavirus. By studying the specifics of this virus and how it attaches to cells, the researchers also gained some insights into how the virus may have hopped from animals to humans. They discovered that a bat coronavirus also binds to the ACE2 receptor, but poorly. A few mutations could have increased the ability of the bat virus to attach to the human receptor, allowing the hop to humans. The researchers also analyzed the structure of the spike protein in pangolins, which could be an intermediate host between bats and humans. They found that one of the pangolin coronaviruses could potentially bind to the human receptor, supporting the notion that pangolins were intermediate hosts of the virus.
That's pangolins, not penguins. The closed capture didn't work quite perfectly. Pangolins being a mammal with scales over it, they're adorable. A penguin, of course, being a bird with feathers. I wanted to show you that just to show that with just a few mutations, those viruses can change and go from something that really doesn't bind very well to cells to something that does and makes that lock and key bind pretty strongly between the virus and the human cell. One of the questions that came up from you guys was, how long does COVID-19 survive outside of a body? And this is from a summary from the New England Journal of Medicine, that the virus can live in the air and on surfaces between several hours and several days. The study found that the virus is viable for up to 72 hours on plastics, 48 hours on stainless steel, 24 hours on cardboard, and 4 hours on copper. And it's also detectable in the air for 3 hours. But one of the caveats that this summary of the journal put on this was, even though they can detect it, the question is the viral load that's on these surfaces large enough to cause an infection? And that, of course, all depends on how much virus started there. But anytime the virus is outside of the body, it can't survive and it's going to start to die off. And this particular virus seems to be pretty stable on the scale of things for viruses, that in that it can survive for quite a while outside the human body, but not forever, just a few 2 to 3 days at the most. So let's get back to these interferons and what are they doing? This is an image from your book, and let's just walk through it together. So here's our virus that's going to enter the cell. So maybe it's COVID and it's entered this lung cell. And it's going to start to replicate, and it does that by using the mechanisms within the cell, the ribosomes and enzymes, in order to make more of itself. When it does that, the cell has a defense, and it has a gene that makes this protein called interferon. And once this gene for interferon gets turned on, this cell is going to produce these red triangles, which are known as interferon. So this is our original host cell that was infected by the virus. It's going to start making this interferon. And what the interferon can then do is it's going to leave the cell through exocytosis, similar to the way a neurotransmitter is going to leave a cell. And then that can be picked up by a neighboring cell. So a neighboring cell would have a receptor for that interferon, and that's going to cause a cascade of events that are going to lead to the DNA in this cell to make some antiviral DNA. And those antiviral RNA are going to make proteins, and those proteins can block viral reproduction. So it's kind of your anti-virus reproduction proteins. So the original cell that got infected, the interferon doesn't help that cell, but it helps their neighboring cells to prevent from becoming infected. Another component of your innate immune system, the inflammatory process is localized, trying to contain pathogens within a specific place. Your fever, however, is more of a systemic response. Your entire body's temperature is going to increase. How does that work? Well, white blood cells that are involved in the innate immune system, so things like maybe your macrophages, for example, are going to release a protein called a pyrogen, and think of like a pyre, a fire. That protein is going to circulate through the body and eventually get to the hypothalamus. So here we've got us looking at a sagittal section of the brain, and in here you've got the thalamus, pineal glands back here, and then you have this hypothalamus and the pituitary glands. Well, the hypothalamus, this is where your body's thermostat is and tells your body where what temperature it should stay at, right? At what point are we going to start sweating? At what point are we going to start shivering? How do we keep our thermostatic homeostasis? Well, when pyrogens are around, it tells the hypothalamus, well, like, let's bump up the thermostat a bit. And there's been big questions. Like, fever definitely seems to improve your body's ability to fight off infections. Big questions about how exactly that works. The original hypothesis was maybe that higher temperature makes it, maybe the bacteria and viruses don't survive well at that higher temperature. There's also some evidence that shows that it's increasing your metabolic rate, and specifically the metabolism and effectiveness of some of your white blood cells. There's specifically some lymphocytes that seem to work better at a warmer temperature, so that if your white blood cells are working better at a higher temperature, then you're going to fight off the infection faster. And your textbook also talks about that the liver starts to sequester iron and zinc, both of which would be needed for reproduction of things like a viruses and a bacteria. So that also might help to deter growth of the pathogens. What's too high? Well, I have this on the slide, but after talking with Laureen today, a high temperature, a high fever is considered to be 103, 104 or so, but that's variable per person and what your age is. That's it. Roll the credits. Just kidding. I have some questions for you. So when comparing virus and bacteria, I'll let you read through that while I wet my whistle. What do you think? All right, if you need some more time to think, hit pause, because I'm going to move forward. Reveal the answer. C, bacteria release toxins. A, bacteria does not need a host cell to replicate, but viruses do. A virus is not a living cell, but bacteria are. And antibiotics work only against bacteria. They do not work against viral infections. But often you can have simultaneously a bacteria and a viral infection in the body at once. That's always fun. All right, phagocytosis is conducted by, all right, pause it if you need more time. Answer is B and C, both neutrophils and macrophages. Natural killer cells are going to cause apoptosis. And neutrophils and macrophages have some other tricks up their sleeves, but they are primarily doing phagocytosis to get rid of cells and debris that we don't need in the body. All right, last one, histamines. Think about it, think about it. Answer is C. So histamines are not released by neutrophils. They are released by mast cells. They're not what causes a fever. Do you remember what does? Starts with a P. Good work. They do increase capillary permeability. They do cause dilation of blood vessels. They do not activate your lymphocytes. That's going to come a bit later. Lymphocytes are important for specificity of your adaptive immune response, but your histamines are not responsible for that one. Okay. Which one? What type of cell is this little joke about? Yeah, natural killer cells. Okay, that's it for now. Next time, next session, we will be talking about the adaptive immune system. See you then.